All right, so welcome everybody. My name is Christian Plessel from the Paderborn Center for Parallel Computing at Paderborn University. And we're hosting today a seminar in the seminar series of the National High Performance Computing Initiative NHR Center for Atomistic Simulation. And it's my great pleasure to welcome Dmitry Morozov, who will give a talk on using novel GROMACS-CP2K interface to perform multiscale QMMM simulation. Let me say a few words to introduce our speakers. Dmitry studied chemistry and received his PhD degree in 2013 from Moscow State University. And then he joined in 2013 the group of Professor Hrunhoff at the University of Jyväskylä as a postdoctoral researcher in Finland. In 2015, he received the Academy of Finland postdoctoral researcher funding. And from 2019, he's also working with the BioAxle Consortium on the implementation of multiscale methods for performing simulation of biomolecular systems. Since his graduation as a PhD student, his research interests are the development and application of hybrid quantum mechanics, molecular mechanics, QMMM methods. And his main object of studies are biological system structures, dynamic and reactivity for both ground and excited states. We're looking very forward to your talk, Dmitry. Welcome.
Yep, thank you, Christian, for kind of invitation and for introduction. So, hello, my name is Dmitry Morozov. So, yes, as Christian already said, I will present today our fastest development of the BioAxle Consortium, our interface between GROMACS and CP2K packages to perform multi-scale simulations. And let me start with a brief outline of my talk. So first, I will maybe not so many people is familiar with what is QMMM and how it could be used. So I will introduce that. Then I will introduce our developments, practically the GROMACS-CP2K interface, how it's implemented and how it could be how it's implemented and how it's working. Then I will show a couple of examples how it can be used to perform simulations of simple systems and of complex protein simulations as well. So, and let's start with the first part. So what is QMMM and why do we want and how can you use it? So basically, when you are talking about modeling of the biological systems, and it's where the QMMM started, basically, and then when you look into the typical biological system model, some protein, DNA, whatever, it usually simulation. You usually perform simulations in the box of water, so in the droplet of water. So typically, your system is much larger than 10,000 atoms, which is kind of a problem for classical quantum chemistry, because classical quantum chemistry, of course, is operates on much smaller scales. So it's typically have like limit to several hundreds of atoms. And without additional tricks, of course. And so it comes the problem that you need somehow to describe this biological system, but you want sometimes to have some non-standard residues or some reactivity, which you cannot achieve with classical molecular dynamics or molecular mechanics. So you need to go to quantum chemistry. And for that reason, there was developed a multiscale approach. So basically, when you combine different levels of theory, to achieve both size of your system and individual resolution of, for example, which is needed for reactivity. Actually, not so long ago, in 2013, when I got my PhD, actually, for this kind of methods, there was given a Nobel Prize to Karpus, Levitt, and Warshall. And basically, their original idea from 70s back, is that you need to describe your system, split your system into two parts, as I already talked, which is called QM part and the MEM part. And the small QM part, then it's basically a region where your chemistry or photophysics or whatever happening. So where your interesting part of your system is part of the system which you are interested in is located. And you want to model this with electronic structure methods like DFT, like ab initio quantum chemistry, and so on. And the rest of the system, you're basically treating at the Forcefield level. And this can be done, of course, very efficiently because it's really cheap method. And also, what you want to do, of course, you want to do something with the system. Yeah, you either want to optimize, do a minimization optimization of geometry, or want to do molecular dynamics, or you want to do some free energy methods. So, of course, you need to integrate the trajectory, update coordinates, and so on. And where is QMM interface? What's called usually QMM interface is something in between, which usually provides connectivity between all these parts, combines, for example, forces from both QM and MM, put it into the trajectory integration, update coordinates, send back coordinates, get forces, and that cycle repeats. So, and QMM interface usually here between these two parts. Okay, so now I want to talk a bit more about the interactions, how you can do this actually, how you can solve different things like quantum chemistry and molecular mechanics. So, and usually it is done like the following scheme. So basically, you first split your system into QM and MM. You just determine which atoms in your system will be QM. Then you know that for all atoms inside the QM region, you are straightforwardly doing the electron extraction methods, DFT, ab initio, and so on. Between atoms in MM subsystem, you're also straightforwardly doing exactly the same thing which you are doing usually. So it means that Forcefield description. And the tricky part comes here. It's actually interactions between QM and MM particles. And for that case, there is at least three main schemes which is now employed in the literature or whatever. So first of all is the simplest one. It's called mechanical embedding scheme, where you basically your QM part is by itself, so it does not feel field of point charges from the MM part, and the total and it's calculated like in gas space, yeah, in that case. But where correction comes in, that you calculate first the whole system as a MM system, like classical MM system. Then you calculate your QM part as a QM system. Then you calculate the same QM part with MM force field, and then you add QM and subtract MM basically. Yeah, basically you replace QM interactions by MM interactions with QM interactions in that case. This is called mechanical embedding because you are doing this on the classical molecular mechanics level. There is more elaborated one, which is called electrostatic embedding, when you include the effect of the quantum wave function polarization due to the point charges from the MM part. In that case, usually you calculate your energy additively. So basically you calculate first MM part without this QM part, like this minus this, and then you add your QM system in the field of point charges. In that case, you have two forces, additional forces. You have forces acting on the QM part from the point charges of MM part. And of course, this force by the third Newton law, yeah, it also should act in other direction on the MM charges. And there is a third embedding method which I use now. It's called polarized embedding, polarizable embedding, where you also include partial polarization of your MM charges due to the QM density. This usually happens when you have a so-called polarizable force field, which are much more, but this method is usually much more expensive and not so widely used now. Yeah, so basically you also include back polarization in that case. Okay, so some of you may ask, but if in case of protein, of course, what happens also, and which is not explained in that schemes, what you will do here when you break covalent bond between the QM and the MM part, if you have a broken covalent bond between the QM part and the MM part. And for that, there is also several treatment methods. So we need to somehow to reintroduce the covalent bonds, especially because from QM part, there will be just an empty valence. So you need to cap this with something. And from MM part, there will be a dangling bond. Yes, so there is at least several schemes. The three main here is shown here. So first is so-called link atom scheme. So I've explained it here. So basically what happens that from the quantum part, you add additional quantum hydrogen or another atom, but in 99% of cases, it will be hydrogen atom with a small basis set, which basically caps, which is located at the typical distance along this bond for CH, for example. For CC bond, you can put it somewhere here. So it's 170, I guess, to 1 ratio, something like that. And then you can cap this dangling balance from the QM part. So there is another approaches which are not so widely used. So for example, you can also cap this free bond with pseudo-potentials, additional pseudo-potentials which act on the QM system, or you can just even put just additional hybrid orbitals on that carbon atom, which will cap this basically broken bond. You reintroduce this bond via the orbitals, additional orbitals. Okay, so next I want to talk when we are typically using this in biomolecular simulations. And here I have 4 at least major fields where it is used now, QMM. So first of all, it's of course a computational enzymology. So when you want to do reaction mechanism inside the enzymes, calculating rate of the reaction, calculating effects of the mutations, calculating substrate activity, for example, and so on. Different substrates, I mean. Then you, another typical is of course the structural biology, when you want to do a refinement of the crystal structures, especially in case when you have a very exotic things like cofactors and so on. Also very closely to that is also a ligand protein interaction. So when you want to calculate the binding energy or specificity of some ligand, which is not standard, and you cannot have a very good, you cannot obtain a very good, for example, molecular mechanical parameters for it very quickly. And fourth way is to do also photobiology. In reality, QMM also works quite well for the excited states. So you can also perform in excited state dynamics using the QMM. That's what I've also done. Yes. So we have another examples of usage if you want. There was a workshop last year where we showed some best practice from the top scientists of the QMM field, how they're using this, if you're interested in that, what else you can do with this method. So, and here you can ask maybe the question, but if this method is so good and it has so wide usage area, so why not everyone is yet using it? So, and last year we committed a questionnaire on the PyExcel website. And we asked basically people, one of the questions was, do you use simulations in your research? And only less than half of the people said like yes. Another approximately half actually said, more than half said like, yes, we are tried, but we had major difficulties, and we are not yet tried, but we are considering, but most may also we want to know what difficulties is. And when we asked what is the major difficulties, which are some hindrance, hindered you from using QMM. The major was not about even the knowing of the QM background theory, but because most of the modern chemists know what is QM or quantum chemistry, at least have some sense of that. But the problem is that when they come to the actual software package, they don't know what to do. I mean, they don't know how to choose a suitable parameters, which will be good for their system. They don't know how to have a good quality of the results, how to check the results. It doesn't make sense or not, and so on. And finally, they want to know, but major just want to have good and relevant practical tutorials, which we are also working on in the PyExcel. But yeah, the main idea is that probably we need to think how to make interfaces simpler to use. And that's where we started to developing our new interface. So, and let me go to the second part, basically when I want to reintroduce you our development in that direction. So basically, in our case, we decided to use interface between CP2K and GROMACS packages for several reasons. But major was that we wanted the whole software bundle to still be free to use for everyone. And the second one, we wanted that our QMM implementation will be fully periodic. Like usually you are doing the molecular dynamics, you are dealing with periodical systems in a box. And for that, CP2K is the best software because they already have the interactions which are periodically implemented periodic interactions because of the of how CP2K works actually. So in that case, for the our total like Hamiltonian of our system, of course, is three components. So first, Forcefield, for which GROMACS is responsible. It's pure MMM interaction. The second one is pure quantum chemical, for which CP2K is responsible. And for interaction of QM and MM, we also used CP2K functionality, which is in that case called GIP, Gaussian expansion of the electrostatic potential, that allows user to have a periodic QM-MM interactions between your QM part and MM point charges. Periodic, I mean, in the sense that it is in periodic pose, in periodic boundary conditions, basically. And for integration and for performing dynamics and minimization, we using the GROMACS. It's the main driver, basically, of this of the integration of the trajectory in our case. So basically, what is Forcefield description in GROMACS? So it is very simple. So you basically have a number of parameters, which are usually called bonded and non-bonded. Non-bonded is usually charge, Lennard-Jones interaction, so on. Bonded is usually vertical, like bonds, angles, dihedral. Yeah, so bonded usually expressed as some kind of harmonic oscillator function or something very similar to it. And non-bonded usually there are two components, so Coulombic interaction and Lennard-Jones interaction. So classical things. And how we integrate this with the periodic, how the CP2K basically handles the periodic interactions, as I said to you, with QMM, they use first so-called GIP scheme. Basically, this is your initial system when you have QM and MM. So what GIP is practically doing, it projects your electrostatic potential from all your periodic MM system onto the grids of your QM system, because it's DFT cost program, you're using some grids to integrate the density. Then you're projecting this and basically get such kind of this picture when you have QM plus projected potential from the MM part. Then in reality, CP2K calculates such kind of QM system with the wrong periodicity, but it is what Quickstep is doing, a CP2K algorithm, quantum chemistry algorithm. But then you want to, of course, have back your periodicity of your full box. Yeah, and for that, you use so-called Blöchel scheme in CP2K, yeah, where you decouple and recouple using the multiple expansion of your density. Decouple means that you first calculate all interactions between, for example, this part and all its neighbors in the neighboring cells. You remove that interactions using the multiple expansion. And then you reintroduce it back with the correct periodicity, basically. You just split your system, and with the correct periodicity, you recalculate them back. This scheme was also proposed in 90s, I guess, somewhere, to restore this periodicity of your system. Yes. So typically, how the workflow is looking like in a QMM modeling is the following thing. So basically, in our case, yeah. So basically, you have some input structure, usually like PDB file or something like that, yeah, with your protein, some initial coordinates. So first, you need to do a system setup. Usually, you're doing this. What you're doing, you are adding benzene residues, deciding on the protonation spots, define the box at solvent, I, and so on. And then you usually, if you're using GROMACS, you perform some kind of MD simulation. Yeah. So you do energy minimization, equilibration, production run, and you will get fully equilibrated system at that stage. But then you want to define some part of your system as a QM. And for that, you need to have the following things in the GROMACS usually. So you defining in your index file or whatever a group of atoms which will be QM atoms. And with several QM parameters, you want to reintroduce, introduce them as a quantum and kind of instructing GROMACS that for that atoms, there should be additional forces calculated by the CP2K. And after that, you can do all kind of QMM calculations, like geometry optimizations, MD, even absorption spectra. So what we wanted also to achieve here is that it should be it should be quite flexible for advanced users. So it should be simple enough for all users of the QMM, but it should be kind of flexible for advanced users, which I want to provide their own parameters sometimes, especially in case of QM calculation. Yes, so this is our interface. So what we wanted to achieve here, and what we achieved already, is that first of all, our interface allows user to automatically convert topology, GROMACS topology from classical MD to QMMM, because as I said previously, there is several things which you need to consider when you're doing that. So we will provide users with a validated set of the parameters which for some very widely used QM methods, especially DFT, some DFT functionals, which will be suitable for most of the biological systems. Our interface is, of course, compatible with any simulation techniques, almost any. It's not compatible yet with FEP, but many of others it is compatible, available in GROMACS, and even supporting the highly parallelizable methods like umbrella sampling, like AWH, accelerated weight histogram analysis, and so on. So how we do topology processing in the interface? So topology, so this is your full system. So what we want to do, we want first to remove, of course, partial charge from QM atoms need to be removed. Then we removed all Lennard-Johnson interaction between QM pairs of QM atoms. So only the one which are between QM and the MEM is kept. We remove all bonds inside which are connected to QM atoms. However, keeping the bond here, for example. We remove all angles which contain 2 or more QM atoms, and the hedrals which contain 3 or more atoms. Also, constraints on the waters will be removed in that case. If some of your waters are fully inside the QM region, then we also remove the constraints from them. And we also generate the CP2K input file, also defining these links atoms here and here. Okay, then I want to talk how usually one implementing these QMM interfaces and how it's looked like before. So usually this is a typical flow of your MD simulation. On the example of MD simulation, yeah. So basically, this is a cycle which continues until you take a number of steps, yeah. And usually when you do some interface, of course, it's looking like that. So on the side of this main cycle, you're always calling some additional functions, which make sure in each particular step that you are doing fine. Yeah, that all quantum system will be described correctly and so on. So what it causes that your QMM code is usually scattered over the whole cycle of the MD simulation. It's really hard to integrate and implement new features or change something because you will need to look where to change and where to hack into the code again. It's really hard to maintain. It's one of the main reasons. It's almost not maintainable, such kind of, because if at some point something changes in the QM package and you need to redo basically everything here, yeah, it often happens, and then you need to cut and redo everything. And it's also a problem of that interaction pattern between the codes. That's why for our approach, we employed a bit different scheme. We call this unified API, which I've pointed here. We call these MDModules in GROMACS. And what these MDModules is doing, basically, they allow external codes to provide forces and energies and setups for the GROMACS in a unified way without scattering the code. In GROMACS, in reality, it is looking like 3 lines in the main code, which can be removed here within 2 minutes, if needed, or added within 2 minutes, and that's all, basically. And how it's looking is looking the following. So basically you have a framework which collects all kind of notifications that are coming from the code. Basically, it could be, for example, if your coordinates change or you want to calculate forces, you basically provide coordinates, and MDModules framework will provide this data further to QMM module like here or any other modules. There is several now in GROMACS, and get back from them individually calculated forces and energies, and uniformly provide this back to the main GROMACS code. So basically now your whole code of the interface is isolated here, and everything else is just managed uniformly through this MDModules framework. So it of course have a huge benefits because you have a less dependencies now inside your main code. Yeah, so like I said, now it's like 3 lines in the main code, which activates your MD module. And of course, your main cycle should not need to know what happens within each individual module at all. Like it's very easy to integrate and maintain, at least from our point of view. And for example, if something stops to be supported, it can be cut out of GROMACS within, like I said, within 1 day, basically less. Or something can be added quite fast also. And of course, we have a predefined interaction pattern, which cannot be changed by the module itself. So they need to follow the rules. Okay, so it's a bit more programmist part of this, my talk. So let us continue then to the some examples. So how the setting up of QMM calculation looks like now. So basically you provide, as I said previously, you have some classical MD already set it up in GROMACS. Then you just put these 5 lines in the MDP file of the GROMACS. So basically this is instructs that your module is active. This is a group from index file with QM atoms. Name of the group. It's basically a method you want to use. This with preset parameters. For now, this means that it's PBE with the double zeta basis set. And also you need, of course, a charge and multiplicity to be known. State, basically spin state. Yeah. So, and interface at the GROM-PP level, it produces you a usual GROMACS TPR file for the simulation, which can be then put as an input for MD-run. And the good thing is that this file holds all information needs for the simulation. So it encapsulates also CP2K input into that. And then you can do QMM calculations. So this is what typically generated CP2K files looks like. So what the interface requires here, that you have basically run type should be energy force, of course, because you want to calculate energies and force on each step. Then we use method QMMM. We setting up automatically the QM box chip and level scheme in case, of course, you are doing the predefined parameters if you use. So this section switches off. In that section, we switch off all MMM interaction calculation in CP2K. So like non-bonded, like Coulombic interaction and Lennard-Jones interaction and so on. And here, yeah, of course, we provide basis set coordinates and so on. Actually, not coordinates, atom kinds mainly, because coordinates is provided via the external PDB file. Why we need to do that? Because we also want, of course, CP2K to know partial charges of all MM atoms here for JIP. And for that, we are using external PDB file with how it's called, extended beta field that contains a point charge. Extended beta field is the one from the PDB file which starts at column 81, I guess, or something like that. And it can usually, it should contain beta factors, but in our case, it contains charges. Yes, and so, for example, that's how it looks like. So basically, you have 2 segments, which is QM and MM. And here you have 0 charges for horizontal QM atoms, but on the MM atoms, you have something like usual point charges. For example, here is HIP3P water, yeah, here. Okay, so let's go to more use cases. So first is a very simple system. It's just basically HBDI chromophore. It is GFP, green fluorescent protein chromophore, but just in water molecule, not in protein, or in water box, not in protein. So this is a system. And for that system, we first tested the energy minimization. So yeah, the energy apparently minimizes, so yeah, it works. And second, we tested also the energy conservation in the NV ensemble of the dynamics. So it's looking like that. And we observed still a fluctuation around 2 kilojoules per mole from the zero. So basically, this is how it's called, difference from the average point in the trajectory. Deviation from the average value of the trajectory. And we still see the fluctuations around 2 kilojoules per mole. As at least from my test, this is caused by the centering procedure, but you cannot avoid this because or else your QM system will sooner or later run out of the QM cell and everything will break. So you need to recenter it from time to time. And this recentering causes small jumps in energy here and there. But it's really too small. I mean, it's 0.001% probably of the total energy. So maybe it's not so problematic. And it's also pretty stable. I mean, I extended this up to 40 even picoseconds and more. It's still like that. Okay, what I also found here is that walls or some potentials at the edges of your QM boxes in CP2K is not very helpful in that case. So I have just removed them at some point because they are causing some artifacts also as well. Okay, and second test is pressure and temperature coupling. So here it's a trajectory again with 1 femtosecond time step, which is quite big actually for QM1. Usually it's used less than 1 femtosecond. But yeah, so you can see the temperature pressure coupling is called. But however, for pressure, I'm not so sure because for pressure, of course, you want to calculate also virial of your force. But for QMMM forces, it's not so easy to calculate the pressure virial, force virial for the pressure calculation. So it's one of the hard things to do. But however, the barostat in practice works. On practice, the barostat works still. Okay, let's go to more advanced setup. So let's say that you are not happy with the standard parameters, what you should do. And for that reason, we introducing a method called input. In that case, you can provide an external CP2K input file to the GROMPP, which GROMPP will preprocess and use as a input, basically as a further input for the calculations. In that input, you can set up almost anything you want. Only limitations is that your runtime still should be force, energy and force. Yeah, and you should use external PDB file for point charges, as in previous case. This is basically restrictions, only restrictions. Yes, and you can do a QM calculation even with external CP2K input, which also will be saved inside the TPR, so it's fully portable implementation. And for that, I want to show a couple of examples in the proteins, for the proteins case. So first is umbrella sampling simulation of the ring isomerization reaction, like from that to that in the phytochrome protein. So basically, this is again the chromophore, but it's a ground state process, which I want to show you. So it just can isomerize in the thermal condition and room temperature from one to another conformation. So basically, what is different here is the position of these two rings called C and D ring. Yeah. And how it's done. So basically, chromophore region and waters and stat amino acids, which are shown here, are QM part. Everything else is MM part. Again, PBE functional, Ember 03 Forcefield. So this is how the reaction looks like. Yeah, like the coordinate of the reaction. For coordinate of the reaction, here we are used angle between the normals of C and D ring, basically. So it's kind of dihedral mostly in that case. And here is the final profile. So here we are used around 20 umbrella windows. Each was simulated for like 20 to 30 picoseconds with 10 picosecond pre-equilibration. at 300 kelvins temperature. And here's the profile. So it's really smooth and good looking. Yes. More interesting case. Of course, this is very simple coordinate. Here we use just the hydral angle, as I said. But what about complex variables? Let's say like in this case. So basically, this is more close to where we are now. I mean, in the pandemic. So basically, this is enzyme called RNA-dependent RNA polymerase of SARS-CoV-2 virus. So basically, because it's RNA-containing virus, it should replicate its own genetic material, which is RNA. And for that, there is a specific enzyme called RNA-dependent RNA polymerase, which replicates the RNA sequence. And it's one of the main targets nowadays for the treatment of the disease itself. Because if you block this enzyme, of course, the virus can no longer replicate itself. So it's a high selectivity and efficiency enzyme. However, it is kind of normal polymerase or so-called phosphoproteins here, which catalyzes the triphosphate ATP, something like how it's called, nucleotide phosphate reactions. Here it's doing, of course, the addition of your nucleotide to the RNA chain, for example, like ATP. In that case, so this is what the reaction happens. So basically, on the right side there is ATP residue, and on the left side there is chain, RNA chain, and the reaction looking like that. The rate limiting step, basically, it's transferring of the phosphor from ATP to the chain, incorporating it to the chain, and then remaining diphosphate will then be ejected of this protein. So this coordinate, and the problem is that coordinate of the reaction that it's really complex. It's not so simple. So this usually when you read the literature about these proteins, enzymes, the reaction coordinate is defined like a difference between two distances. So it difference between distance from this phosphorus atom to this oxygen atom of the ATP, and distance between this phosphorus and oxygen atom of the previous nucleotide already in the chain. And of course, for that, you need to use this complex variable, like collective variable, yeah, which are difference between two distances, and scan over that. In that particular simulation, I have used plant integrated into the GROMACS. But in reality, from the GROMACS 2022, we will have already inside the GROMACS such kind of complex pooling coordinates. So there will be a transformation, so-called transformation coordinates, where you can use any arbitrary mathematical expression to which you can pool also. So something like that will be also possible to done purely in GROMACS. So for that kind of system, we used the following setup. So kind of really big system. It's around 300,000 atoms in total. But actually, we have achieved kind of very nice performance, almost 6 picoseconds per day with 1 femtosecond time step, using for 2 AMD ROM standard nodes, which are kind of standard nowadays for the supercomputers. And in total, we produced around 10 picoseconds equilibration run plus 20 to 30 picoseconds production run, and 6 umbrella windows. So, and in that study, we also, I have also compared performance of the our GROMACS-CP2K approach and the conventional CP2K approach for the QMMM, which is inside the CP2K itself. So basically, this is a distribution over the all umbrella windows. So on the top is our interface, on the bottom it's CP2K, and yeah, it's kind of similar. Yeah, it's fully similar almost, not distinguishably. Yes, but if you look into the profiles itself, up to TS seems like everything is almost the same. But on the product side, GROMACS is a bit lower. So I've probably it's not enough sampling have been done in CP2K, but because for CP2K we've done a bit shorter trajectories than for the GROMACS-CP2K. I suppose that this is due to that fact. So products can be even lower here. So, but conclusion is that, yeah, results are pretty comparable and almost indistinguishable. So it's less than kilojoule, kind of 2 kilojoules per mole here at the end. And between substrate up to transition state, there is no difference like at all. Okay, I guess that was the last slide almost. So there is some kind of further information. So when to expect this interface? So it should come with GROMACS 2022 release, at least it's planned for now. But more information can be always looked into our official sites. We also prepared some best practice guide with choosing the simulation parameters, particularly for CP2K in case of biomolecular systems. It's located here. And we have also several tutorials and talks about this on our BioXcel YouTube channel, which one can also check if you want. So there is some acknowledgments with the people who are working with. So this GROMACS KTH team, which allocated the KTH, which helped a lot. And there is our collaborators. And yeah, thanks for your attention. So basically we are working for the BioXcel. And yeah, now you can ask any questions.
Yes, thank you very much, Dmitry, for a very interesting talk. I would like to stop the recording now before we go into the Q&A part.